Good morning, everyone. Thanks, Laurie, for the introduction. I'm Tara. I'm a postdoctoral research fellow in the Department of Pediatrics. Thanks for the opportunity to chair this session today. And it is my immense pleasure to welcome all the participants as well as attendees for today's meeting on the topic lipids in inflammation. The first speaker of the session is Sanjay Varikuti, who is a research associate at Ohio State University. He is working on projects involving regulation of immune responses during leishmaniasis. His topic of presentation today is Pentalinonsterol exhibits the immunomodulatory action in macrophages through activation of phospholipase A2. This work is carried out under Dr. Parinandi, and I welcome you, Sanjay. Over to you.
Hello. Hello.
Hi, Sanjay. We can hear you.
Okay, thank you. I'm sorry. Hi, my name is Sanjay. I work in infectious diseases research, and I'm from Department of Pathology from Ohio State University. Today I'm going to talk regarding some interesting immunomodulatory molecule which we isolated through the natural compound, and I mean a plant compound. This is a Pentalinonsterol, and give me a second. I'm going to open my share screen. Okay, think you guys are able to see that right now.
Perfect.
Yes,
okay. So it's about the Pentalinonsterol exhibits the immunomodulatory action through activation in the macrophages through activation of phospholipase A2. So basically, the main idea of doing this is I work mainly in the infectious diseases. So we work in the disease called leishmaniasis. So this leishmaniasis is a cluster of diseases caused by Leishmania protozoan parasite. It spreads around like 80 tropical and tropical countries and affects around 12 million people, leading to the second largest cause of mortality. Presently, there is no vaccine and no FDA approved drug. So we, in order to develop, in the process of developing some new drugs, we also work on some natural compounds. Few years back, so we come across with the plant called Pentalinonandroxy, which is from Mexico. One of our research associates and postdocs, they bought the root of the plant. So we started isolating the pure compounds from it and then testing out of these pure compounds. So he found out like the Pentalinonsterol root extract has some anti-leishmanial activity. So based on it, we isolated some pure compounds. So one of the pure compound is a Pentalinonsterol. It's a sterol molecule. So it's a natural product isolated from that roots. And our previous studies have shown that this root extract, Pentalinonsterol, exhibits immunomodulatory properties which markedly increase the activity of macrophages and dendritic cells, and which can be seen by increasing the NF-kappa B activation and all those. So next, given the beneficial characters of this pen, so the objective of the my study is to illustrate the mechanisms by which this pen can modulate the immunomodulatory property, exhibits the immunomodulatory properties. So one of the membrane phospholipid is a phospholipase A2, which actually involves in the homeostasis, cell signaling, generation of the eicosanoid inflammatory bioactive lipids. So here we want to investigate whether this pen would modulate the activity of PLA2 in raw macrophages or also the bone marrow derived macrophages from the mouse, or it can activate the upstream PLA2, upstream phosphorylation and all those things. So basically our hypothesis to test the pen changes the PLA2, activates the PLA2 because it is present in the membrane phospholipids, and That in turn activates CPLA2 activation, hydrolysis of membranes, and then release of the arachidonic acid, which in turn breaks down by COX and LOX, lipoxygenases and cyclooxygenases, and release the eicosanoids. That induces the cytokine synthesis and also the primary immune responses, like mainly the Th1 immune responses. That is a proposal we want to test in this. So to test that, we took the raw macrophages and we treated them with Pentalinonsterol with the different doses. As you see, with the increase of the doses as well as with the increase of the time, Pentalinonsterol actually induces the arachidonic acid. That is the testing method we can actually test like it is activating the PLA2. So melittin also, we tested a drug called melittin, which is actually a natural inducer of the cell mechanism. So we tested that with the pen as well. So that also acts as the same way as a pen, which is a known mechanism. So the reason of testing it is to have a positive control kind of thing. Next, so we found out Leishmania donovani, it's a parasite, so also induces the PLA2 here in raw macrophages by increased parasitic doses as well. So, and also we tested like what happens if we put the parasites and pen together. So we see like pen enhances the L-donovani induced activation of PLA2 in raw macrophages as well. So if you see the pen in 30 micromolar concentration here in the parasites as well, and by combination of that, it is significantly increasing. So next we want to check whether this pen modulation or release of the PLA2 or release of arachidonic acid is a calcium dependent or calcium independent, since the calcium is a main signaling, main molecule which involves in these phospholipid metabolism. So we tested the BAPTA, which is intracellular calcium chelator, and also extracellular EGTA. So with the BAPTA, if we stop the calcium, intracellular calcium, significantly it is inhibiting the release of the arachidonic acid, but not the extracellular like EGTA. And then we want to test this, is it a cytosolic modulator or not? So we tested the BEL and as well as a MAFP, which is specifically the secreted PLA2 inhibitors, and it seems like the pentalino and melittin-induced phosphorylation release of arachidonic acid is not decreased by BEL, but significantly decreased by MAFP, which is indication of like it is cytosolic, depend on cytosolic calcium as well. Due to that reason, so we selected the CPLA2. We want to test this since this is cytosolic PLA2 inhibitor is working. So we want to test on this as a specific of CPLA2 inhibitor. So if we use a specific CPLA2 inhibitor, both pen plus melittin-induced arachidonic acid release is significantly decreased, and also pen and parasite-induced arachidonic acid release also significantly decreased by CPLA2 specific inhibitor. So we tested this in in vivo method. So we took the mouse, which has a wild type as well as a CPLA2 knockout mouse, and then we tested the same thing again in the in vivo system, whether CPLA2 inhibits. So this is showing that wild type and CPLA2 knockout mouse, because it's and now here if we see the CPLA2 knockout mouse and compared to the wild type, by induction, so we can see the control and parasites and pen, we see higher amount of arachidonic acid release, but in the CPLA2 in knockout mice, it is significantly decreased. So which in turn tells that this pen-induced arachidonic acid is completely dependent on CPLA2. So next we tested this, is this a CPLA2 or the arachidonic acid release is the depend on upstream ERK or ERK phosphorylation or not. So here we check these in 15, 30, 60, 75 minutes, and it significantly depend on, it's clearly shows that this is clearly depend on the ERK phosphorylation by, this is a densitometric analysis, and you see that. And next, we also tested this pen inducing the serine phosphorylation of CPLA2. Again, Same thing, Western blots, we check the densitometric analysis clearly shows that pen and melatonin induces CPLA2, phosphorylation of CPLA2. The same thing here, the pen inducing the translocation of CPLA2 in BMDMs. We tested this again in the microscopic way. Basically the control and Pentalinonsterol and pen and melatonin, which is a CPLA2, clearly showing by the microscopic method. Last thing we want to check whether this pen induced CPLA2 results in a release of inflammatory cytokines. So we tested the inflammatory cytokines like TNF alpha and interferon gamma, basic Th1 immune responses. With increased concentration of the pen, we did see an increased concentration released of TNF alpha by BMDMs as well as Ra macrophages. And by using a CPLA2 inhibitor, this TNF alpha is clearly decreasing. So that in turn tells clearly the pen-induced arachidonic acid is CPLA2 dependent, and it induces the immune responses. So basically, results demonstrates that pen activation results in dose and time dependent manner. It induces the activation of PLA2 and depend on specifically the CPLA2. Taken together, we can demonstrate that pen exerted its immunomodulatory actions through activation of CPLA2 and macrophages, in which potentially use the development of adjuvant for vaccine against leishmaniasis. That is the end of my presentation. Take any questions.
Thank you, Sanjay, for the interesting presentation. And there is time for a few questions. Susanna has asked, like, cytoplasmic PLA2 co-localize with COX2 in your system?
We did not check that. Basically, we want to check only how it is going through the arachidonic acid release or PLA2 and CPLA2 only, but we didn't go through the icosanoid mechanisms until now. We did not check that.
Yeah, she also had a question like, pen, does it act only with cytoplasmic PLA2 isoform?
Yes. Yes, we did check this CPLA2 and secreted CPLA. I did not show the presentation because of the time lag, but yes, it only acts through the CPLA2 mechanism. Yes.
Oh, you checked the secretory PLA2 as well, right?
We did. Not much different, but we did find that CPLA2 specifically inhibiting. So that is the reason we went through testing in the knockout mice and as well as using a specific inhibitors of CPLA2.
Fatima has a question like, what is BEL again? Like,
so it is BEL is so let me it's a MAP kinase inhibitor basically. BEL, give me a second. I think I forgot the name of the BEL, but technically it's an inhibition. We are checking here is the PLA2 inhibition, specific inhibitors of the MAP kinases.
And Shamroop has a question like, have you tried any agonist or antagonist to cytoplasmic PLA2?
Cytoplasmic PLA2 antagonist or agonist. Specifically inhibitor only we tested. I did not test any specific antagonist or agonist. Yes.
And Emily has a question like, how do the amounts of PEN you used compare to what a macrophage might see in vivo?
So macrophages basically, as I said, this molecule never actually used by anyone. So ours is the first lab who got this molecule. So anything what we are testing is like do and testing purpose only. So we clearly did not know how much the macrophage can take it or how much it can handle it. But as we have published like almost 6 manuscripts on this Pentalinonsterol, and every time we test these, the the cytotoxicity of the Pentalinonsterol, and it is very less compared to any other drugs. And we publish the data in immunomodulatory effects of the Pentalinonsterol in this manuscript. To actually answer exactly the question, like how much the macrophage can take those, but I don't know. But because we did not test that, we only tested the cytotoxicity of that.
Okay. And Ritika has a question, like why did you choose only TNF alpha? And is it because it plays a major role in Leishmania
Yes,
infection?
obviously. So TNF alpha is one cytokine which will be released by any of the cells. That is the first immune response, right? When the dendritic cells in like IL-12 induce the dendritic cells and then release, activates macrophages. Macrophages release the interferon gamma, TNF alpha, nitric oxide. So that's how the process of immune reaction starts. But basically, whenever there is an inflammation or induction of immune responses, the first we would see the TNF alpha very easily compared to any other cytokines. So we could I tested the interferon gamma as well. Since this is not infected macrophage, so they don't release too much interferon gamma. But the TNF alpha, yes, it's cytokine which you can see by any cell modulation.
Okay. I think you are almost running out of time, Sanjay. And one last question. What is the, how does the pen act through? Is it through steroid hormone receptor? The action of pen, entry of the pen to the cell?
That's a good question. So we don't know the answer yet. So I know the mechanism how it is acting right now because of the study, but how it is uptaken by the cell, since it is a steroid molecule, cholesterol molecule, basically it has a hydrophobic molecule, right? So it may be uptaken by the cells because of the phospholipase bilayer will take those molecules. But specifically, what is the mechanism through which we need to test? Still need to test.
Okay. I think we have to end this talk now. Thank you so much, Sanjay, for the interesting presentation.
You're welcome. Thank you.
Yeah. I think you have to stop sharing the screen. Yeah.
Sorry. Okay.
So let us move on to the next speaker of the session, William Bresette. He's a PhD student with Dr. Floyd Shilton in University of Arizona. He will be presenting his work entitled In Vivo Metabolomic and Lipidomic Response of Healthy Humans to Intravenous Lipopolysaccharide Challenge. Welcome, William. Over to you.
Thank you. My name is William Bresette. I am a student at the University of Arizona, first year, now second year PhD student. And we used a metabolomics method to measure the response to lipopolysaccharide challenge in humans. So our study design is we took lipopolysaccharide at Penn State University, and 9 people were injected with 0.6 nanograms of lipopolysaccharide per kilogram into 10 healthy adult males. And as we can see, lipopolysaccharide is a 3-component outer membrane part of gram-negative bacteria. It has an O antigen, a core, and lipid A. And the lipid A is the main driver of the inflammatory response. Okay, and this is vitally important to understand this mechanism or the metabolomic response in vivo, because sepsis and infections are huge killers in the world, and they drive a whole bunch of other different diseases as well. So one of the big ones going on right now is coronavirus, and that is a viral infection, but here we measure a gram-negative bacterial infection, which also kills a lot of people every single year. So the main mechanism of uptake for lipopolysaccharide is through the TLR4 mechanism and to promote NF-kappa B. And this NF-kappa B then goes on to produce pro-inflammatory mediators, including cytokines and TNF-alpha. And these are short-term responses, acute responses, and CRP increases a little bit later. So our study population, so we tested 10, and then nine were used in the metabolomic analyses. And they were all healthy males, aged about 27 years old, whose BMI was in the healthy range, slightly overweight, but they had normal blood pressure, normal glucose and insulin responses, and normal cholesterol and lipid panels. And so these were healthy adult males. And so they had a blank slate. And this is testing the inflammatory response on a blank state. And so we measured cytokines using ELISA, and we measured IL-6, TNF-alpha, and CRP. And then metabolites, we used a methanol and water extraction with Avanti polar internal standards for relative measurements on a Waters time of flight liquid chromatography mass spectrometry. And we used an untargeted method. And the metabolomics were measured at Colorado State University. And so how we identified these methods was we pre-selected the compound matches, and then the compounds were adjusted for their closest internal standard. We measured we did significance testing with an FDR correction on each compound, and we identified 248 significant compounds using the repeated measures ANOVA. And then going back to these 248 significant compounds, we went and looked at each spectra data, and we matched the compounds with what should be. And so the unidentifiable compounds were discarded because we didn't know what they were. And the compounds were readjusted for internal standards. And we redid the statistical analyses and found 196 significant metabolites after FDR correction. And then we did many different multivariate analyses to show some data visualization. And so the cytokine markers, we see TNF-alpha and IL-6 having peaks at 2 hours and CRP having a peak at 24 hours. And this is from baseline value. Some notable significant metabolites are 47 phospholipids, 45 increased and 2 decreased, 36 lysophospholipid, 35 increased and 1 decreased, 20 sphingolipids, in which 15 increased, 3 decreased, and 2 increased and decreased depending on the time scale. And there were a few other notable metabolites that were increased significantly due to this response. And this was 15 methyl palmitate, pristanic acid, an endocannabinoid 2-AG, glycerophosphocholene, and 18 hydroxycorticosterone. So sparse partial least squares discriminant analysis, it is like partial least squares discriminant analysis where it takes the top compounds and separates them. And so we can see here that the 0 hour mark is the baseline, and all of these group together quite nicely. And the 2 hour, the 4 hour, the 8 hour, and the 24 hour, they're quite similar. And the 72 hour is significantly different than all of them. So we can look at the variable importance and projection scores of each of the metabolites that are driving this separation. And we can see that 15 methyl palmitate and 5-methylcytidine are the greatest with VIP scores close to 3. And these are all the VIP scores of metabolites that have a VIP score of greater than 1.5. And we can see a lot of phospholipids and other lipids here. So in a pathway analysis, we see that glycerophospholipid metabolism and sphingolipid metabolism are the two main pathways that appear using a KEGG analysis. And the other pathways were only identified with one compound. So the IDs of these were left out, but the two primary ones were glycerophospholipid metabolism and sphingolipid metabolism. And so in glycerophospholipid metabolism, we see a large increase in phosphatidylserines, phosphatidylethanolamines, phosphatidylcholines, and their products of lysolipids, diglycerides, and choline, and glycerophosphocholine. And these enzymes, we did not measure the enzymes, but phospholipase A2, phospholipase A1, phospholipase C, and phospholipase D all have previous reports of increases during an inflammatory response. And so we think that these are the biggest drivers in the inflammatory response, especially the phospholipase A2 reaction. The sphingolipid metabolism, we see that ceramides and sphingomyelins, and especially N-acetyl neuroaminogalactosyl ceramide, as the primary drivers in the sphingolipid metabolism, and with sphingonine and phosphatidylethanolamine ceramides, which are bacterial products, they decrease in the time course. So there are a few limitations to the study. Cytokines and C-reactive protein resolve, but many metabolites can continue to rise. And we're not sure if this is because these metabolites are anti-inflammatory and are helping to solve this inflammatory response, or if they continue to drive the inflammation after more than the cytokines and C-reactive protein tell us. So sample preparation also favored non-polar metabolites over polar metabolites, and internal standards were only non-polar lipids. So we did not adjust for any polar lipids, and the lipids increase in how they increase in plasma is quite unknown currently. One neat thing in the world today is the coronavirus and metabolomics. Now, there's a paper published in May in Cell about coronavirus and metabolomics, and they found a lot of the same compounds that we did, which were the lysophospholipids, as shown here, but they decreased with severity. So I thought that was pretty interesting. And I would like to give a special thanks to my collaborators at the University of Arizona, Colorado State, Penn State, and my lab, the Chilton Lab, here at Bio5 at University of Arizona. And I will take any questions.
Thank you, William. Thank you for the knowledgeable presentation. So there is time for a few questions. So Tara has asked, like, how did the dose administered compare to sepsis patients or those with other diseases?
So we measured just on a blank state. So there's one paper published in 2017 that studied, or that looked at the differences in humans injected with lipopolysaccharide versus sepsis patients, and there were very, very similar responses, but they did not look at the lysophospholipids and sphingolipids like we did. So the responses seem very similar, especially in gram-negative bacterial infections in sepsis patients, with an increase in lysophospholipids and sphingolipids.
Mm-hmm. Naren has a question, like, which bacteria was LPS most similar to?
E. coli. We got the LPS from E. coli.
And Jamie has a question, like, what column was used for LCMS?
It was a Waters column. I can't remember the exact diameter or length of the column.
Mm-hmm. And the follow-up question is, like, did you try to use matrix to help with the identification? Instead of disregarding unidentifiable compounds, you can do tentative identification using matrix. This is also a suggestion kind of thing. It enlarges your metabolome for PCA analysis. For your pathway analysis, what was the total number of metabolites submitted?
Yeah, so we measured 1,007 metabolites, and we did look at the database for identifying the unidentifiable compounds from our proprietary database, but we're not extraordinarily confident or extremely confident with the results that we got and the compounds that we listed here. We are very confident that they are actually the compounds that we see, because we have a very high resolution machine, and the ones that weren't identifiable weren't also not extraordinarily identifiable using that software.
Zaina has a question, like based on which time point did you perform the KEGG analysis?
We performed the KEGG analysis on all the time points. And so if we go back here, the red is an increase. And so the KEGG analysis, we looked at if any of the time points increase from baseline after FDR correction, then we use those metabolites in the KEGG database. And so we can see in these pictures that many of the metabolites increase. And so we just use those in the KEGG analysis.
Maverick has a question, like compared to cytokine profile after LPS injection, how the lysophospholipid dynamics looks like. Does the lysophospholipid the consequences or cause of cytokine production?
So we did not look at the specific function of lysophospholipids in the production of cytokines. We just looked at the response of the metabolome to lipopolysaccharide injection.
Okay. Hamid has a question, like why did you limit the sample size to 9 healthy subjects?
We limited it to 9 because of the cost of running the metabolites, and also there were a few subjects that were tested, but they did not seem to have a very significant response, and so we excluded those. We excluded those from the results.
Okay, I think we are running out of time. William, thank you so much, and thank you the attendees for the thoughtful questions.
Thank you.
Sanjay?
Yes. Hi, this is Sanjay, and now the next is Sudhadevi, and she's going to talk about presentation in a few minutes.
Good morning, everyone. I'm Tara. I'm postdoctoral research fellow in University of Illinois at Chicago. So today I would be presenting my research on the role of sphingosine kinase inhibitor, that is PF543, on modulating hyperoxia-induced airway remodeling. To introduce you to the topic, bronchopulmonary dysplasia is a neonatal chronic lung disease which is characterized by impaired lung development. This is caused as a consequence of the life-saving strategies to the preterm babies with an incidence of up to 68%. So it is estimated annually worldwide about 15 million babies are born preterm, of which 2.4 million babies are born extremely premature, less than 32 weeks of postmenstrual gestation age. So BPD is multifactorial, of which inhaled oxygen therapy and mechanical ventilation are the significant contributors. And this excessive supplementation of oxygen is called as hyperoxia, and hyperoxia-induced bronchopulmonary dysplasia leads to airway remodeling and followed by airway hyperresponsiveness. And the most important aspects of airway remodeling are smooth muscle hypertrophy.
Excuse me, Tara, can you share your screen, please? Could you share your screen, please?
Yes, sure.
Thanks.
I can continue, right?
You may want to start, if you need to start over, go ahead.
Oh, I'm sorry.
That's okay.
And please, there you go.
Mm-hmm. So to introduce you to the topic, bronchopulmonary dysplasia is a neonatal chronic lung disease which is characterized by impaired lung development, and it is caused as a consequence of the life-saving strategies to preterm babies with an incidence of up to 68%. Annually worldwide, it is estimated that about 15 million babies are born premature, of which 2.4 million babies are born extremely premature. So among the risk factors for bronchopulmonary dysplasia, inhaled oxygen therapy and mechanical ventilation are the most common ones. And this excessive supplementation of oxygen to the system is called as hyperoxia. And hyperoxia-induced bronchopulmonary dysplasia leads to lung inflammation followed by airway remodeling, which is followed by airway hyperresponsiveness. The important aspects that we look in airway remodeling are smooth muscle hypertrophy and peribronchial fibrosis. So the survivors of BPD, they suffer from lifelong respiratory morbidities, including asthma, wheezing, pulmonary hypertension, fibrosis, etc., in addition to cardiovascular as well as neurological problems. So the current treatment modalities for BPD are ineffective, and therefore research efforts at clinical, translational, as well as basic levels to combat the disease is very important. Sphingosine kinase, S1P signaling in bronchopulmonary dysplasia. Sphingosine 1-phosphate is a bioactive lipid molecule which is involved in a number of cellular responses, and S1P is produced from sphingosine by sphingosine kinase 1. So PF543 is a sphingosine kinase 1 inhibitor which prevents the conversion of sphingosine to sphingosine 1-phosphate. So in literature, it is already known that the altered levels of S1P is observed in a number of lung diseases, including BPD. Also, earlier studies from our laboratory showed that sphingosine kinase 1 mutant as well as inhibition of sphingosine kinase by PF543 in mice conferred protection against hyperoxia. So, as we know that sphingosine kinase 1 mediates neonatal hyperoxia-induced lung airway remodeling in adults, my focus of study is to look at the impact of PF543 treatment on airway remodeling in neonatal mice exposed to hyperoxia, followed by lung development into adulthood. To brief you the methodology, tracheal aspirates were collected from term as well as those meeting the criteria of mild, moderate, and severe BPD disease. For in vivo experiments, C57BL6 mice were used, and PF543 was given at 5 milligram per kilogram body weight intraperitoneally from postnatal day 3 till postnatal day 10 during hyperoxia. And then the animals were sacrificed either at postnatal day 10 or they were allowed to grow in room air to become adults for 4 weeks, and the animals were then sacrificed at PN 56 for bronchoalveolar lavage collection, histology, or lung function studies. So in vitro, sphingosine kinase was inhibited either using mutant plasmids or by inhibiting with PF543, then followed by 95% hyperoxia, and then the cells were characterized for specific marker expression. So coming to results, so it was seen that S1P measurements using LC-MS-MS, the patients with established severe bronchopulmonary dysplasia had significantly elevated levels of BPD as compared to mild or term normal controls, suggesting that S1P could be a significant contributor to bronchopulmonary dysplasia. In vivo studies with neonatal mice showed that the hematoxylin eosin staining, this is from postnatal 10-day-old neonatal mice lung tissue, and it can be seen that there is improved alveolarization upon treatment with the sphingosine kinase inhibitor PF543 as compared to hyperoxia group. So the objective assessment of alveolarization was done by measuring the mean linear intercept, and this showed that PF543 reduced the mean linear intercept drastically upon PF543 treatment. There was also a significant reduction in the BAL protein concentration, BAL cell count, as well as hydrogen peroxide concentration, indicating protective effect of PF543 on lung inflammatory responses. The plasma S1P levels and plasma dihydro S1P levels are also significantly less in the PF543 treated groups. So as these neonates were grown into adults, the hyperoxia-induced lung damage was obvious, and PF543 treatment as neonates showed protection upon growing into adults. So this is confirmed with mean linear intercept measurement, and BAL protein concentration and cell count also drastically reduced, showing there is decrease in lung inflammation upon PF543 treatment. Then we looked at peribronchial fibrosis, which is a common phenomenon in bronchopulmonary dysplasia. In neonatal mice exposed to hyperoxia, we did not see a peribronchial fibrosis, but as they grew into adults, they developed significant peribronchial fibrosis, which is seen blue here upon Mason's trichrome staining. And PF543 treatment drastically reduced the peribronchial fibrosis as seen in this figure and quantitatively too. Then we looked at smooth muscle hypertrophy by staining with smooth muscle myosin heavy chain, and it can be seen that the treatment with PF543 drastically or significantly reduced the area of smooth muscle actin staining as compared to the hyperoxia treated groups. PF543 also significantly improved lung function. So respiratory system resistance is the total lung resistance, and this is Newtonian resistances, the airway resistance. It can be seen that upon methacholine challenge in hyperoxia treated groups, which is marked orange, there is increase in lung resistance, which is significantly reduced upon PF543 treatment, which is marked red in both cases, showing the protective nature of PF543 even on lung function. Then the neonatal hyperoxia was associated with increased SPHK1 expression in adults, which again got reduced by PF543 treatment. Then we looked at epithelial mesenchymal transition, which is commonly there in SPHK1, which is commonly seen in bronchopulmonary dysplasia, and it can be seen that the ecadherin expression. Ecadherin is a marker for epithelial cell, and epithelial marker ecadherin drastically reduced in hyperoxia, and this reduction was prevented upon treatment with PF543. So to confirm the EMT-like changes in vitro, we used small airway epithelial cells, and it can be seen that the hyperoxia-induced degradation of ecadherin was reverted upon treatment with PF543. Also, vimentin, which is a mesenchymal marker, which got increased upon hyperoxia treatment, also decreased drastically upon PF543 treatment. PF543 also reduced the increase in mitochondrial ROS caused by hyperoxia. So then we treated the transfected cells with SPHK1 dominant negative mutant, and we found that this treatment decreased the hyperoxia-induced mitochondrial ROS production. Mitotembo, which is a mitochondrial ROS scavenger, again decreased the hyperoxia-induced increase in mitochondrial ROS production. Mitotembo also inhibited hyperoxia-induced changes in EMT, as seen with degradation of ecadherin as well as increase in vimentin. So indicating the role of SPHK1 in decreasing mitochondrial ROS and decreasing EMT-like changes. To summarize, SPHK1 inhibition during neonatal hyperoxia mitigates the long-term consequences of hyperoxia-induced airway remodeling. PF543 inhibition of SPHK1 diminished mitochondrial ROS as well as EMT caused by the hyperoxia insult. PF543, by modulating airway remodeling and airway hyperresponsiveness, played a potential role in alleviating BPD-like symptoms. I would like to acknowledge NICHD grant provided to my PI for funding, and I would also like to thank ASBMB for the opportunity given me to present my work here. Thank you so much.
Thank you, Tara. Now I would like to take some questions from the viewers here. So Linda asking that how are the ceramide levels after inhibitor administration?
We did, earlier, like in the study, I haven't shown, but the ceramide levels are also decreasing.
Okay. Second question, Naren asking how was the inhibitor administered? Is it expected to see a reduction in circulating DH sphingosine levels?
Yeah, we did find a decrease in dihydro S1P levels, and the drug was administered intraperitoneally.
Okay, thank you. Now, Catherine is asking, like, are genetic polymorphism in sphingosine kinase that are associated with severity of BPD in humans? Are genetic polymorphism in sphingosine kinase are associated with severity of BPD in humans?
Yeah, there are genetic polymorphisms are that are reported, but in general, I think hyperoxia is one of the main reasons for this change.
Okay, thank you. Sam Roop asking, what is the status of SPHK2 in these cells?
Yeah, it was shown that SPHK2 doesn't have a role to play in BPD.
Okay, thank you. Next question is Nancy. Might a mechanism of action of a sphingosine kinase inhibitor in improving BPD be by protecting lipid raft formation?
Yeah, in this study, like we are saying that it is through reduction of mitochondrial ROS, which further alleviated the EMT transition, and this is the reason for protection of PF543. EMT-like changes as well as decreased mitochondrial ROS production.
Okay, thank you. Next question from May Pone is asking that what is the method to measure the mito-ROS production?
It is through using mitosocs. It is a fluorescent dye. We treat the cells with PF543, or we subject it to hyperoxia and treat it with mitosocs. It measures the mitochondrial ROS.
Thank you. I think there is no open questions right now. I think you have answered all the questions. If there are more questions, you can reply them through chat box, I guess.
Yeah, sure.
Thank you, Tara.